Hello, everyone. Welcome to today's Gates Institute seminar series. We are very fortunate to have one of our own, Dr. Igor Kogut, who is an assistant professor in the Department of Dermatology, as our speaker today. Igor is originally from Ukraine. He received his BS and MS degree in biology and chemistry at the Senescent National University. He then came to Colorado and received his PhD in 2010 in the Department of Biochemistry and Molecular Genetics. He then did a postdoc with John Cambier in immunology. And then in 2012, we were very fortunate to have him join us in the Gates Center and the Department of Dermatology, where he, in collaboration with his long-term partner, developed our combined gene editing and reprogramming technology, which was recently patented. In addition to working on our team project of developing an IPS-based therapy for epidermolysis bullosa, Igor has been interested in developing his own project, which is focused on somatic cell senescence and ways that you might be able to rejuvenate senescent cells. So, Igor, we're really pleased to have you today, and we look forward to your presentation.
Thank you very much, Dennis, for that nice introduction. So today I'm going to speak about one of the projects in our combined lab with Anya. It is human somatic cell senescence and rejuvenation. So several things I would like to say. So at any time, please stop me and ask questions if something gets complicated. And I would like to keep order of my slides in more chronological order. So just going and I think I'll try to explain what is the reasoning behind it. And again, if something gets a little bit complex, please stop me and ask questions. Also, we got 2 Grafts awards with Anya, and one in 2015. This was for IPS technology and like ways to improve IPS technology. Second one, which we just got award last December, it's about this rejuvenation technology, some specific aspect of this, but I think that this technology is way more applicable to way more different fields and everything else. So please let me start with, with a disclosure. So we filed a patent for this technology several years ago, and inventors on the patent, me, Anya, Nicole, and Sean McGrath, who worked with us back there. Also, in one specific slides, I'm sorry now, so I'm not going to tell all factors. So I would like to show most recent data we have, and that seems very exciting, but I just cannot tell all factors we're using in our final cocktail, RNA cocktail, because we're planning to submit additional continuation in part for this patent. So until it's submitted as a patent, I cannot tell this data, like full data yet. Okay, please let me start. So in the first part, I would like to talk about developing RNA-based cocktail for the functional rejuvenation of human somatic cells. And we can start with aging. So aging is a progressive degenerative state, and very briefly and very simply, it's a tissue stem cell depletion, tissue inflammation, matrix alteration, and very important, cellular senescence. So it's not identical processes, cellular senescence and aging, but they're interconnected and quite similar. One of big problem for translational medicine that in transplantology, if you try to transplant tissue from aged person to another person, quite often it's not working pretty well. It's in case of skin, I mean, again, it's a problem because you cannot transplant skin to another person. You have like it's only for the same person, but it's difficult to expand it, especially if it's aged person. In case of many organs, engraftment rate also is pretty bad if it's taken from aged person also. After my conversation with Dr. Terry Fry and with Elizabeth Franks too. So I think you're an audience. It's a can be problem for CAR T cell therapy as well, because if you take cells from, again, aged person and try to expand them, they might not expand enough or might be not functional after this expansion. And again, very interesting notes I got that tumor infiltrating lymphocytes also have eroded telomeres, and again, they cannot perform very well as this. I think our methods can address it, but this is our future work. Okay. If you're talking about cellular senescence, we can see changes in mitochondria, epigenetics, DNA repair, and quite often it's connected to telomere dysfunction. So telomeres, again, this is the protective ends of chromosome, and I'm gonna talk a bit more next slide. But through different pathway, they're connected to many aging like phenotypes and issues with cells. Like, for example, mitochondrial dysfunction can be connected through activation of p53 and PGC1 alpha and beta. Also, stem cells exhaustion. Epigenetic regulation, quite often connected to sirtuins. Senescence itself through p53, p21. And again, it looks like, I cannot say that everything in aging senescence connected to telomeres, but it's close to this, I mean, in my opinion. So what are telomeres? So we have linear chromosomes, they're not circular bacterial, and ends of chromosomes are protected by telomeres. This is a short repeating nucleotide sequences. You could see it here, TTA, GGG. And they're quite long. I think in a human, it's about 16, 20 kb, like when, like for neonatal cells. However, the implications that machinery which duplicate those chromosomes cannot fully duplicate telomeric sequences. So the small pieces chopped each division. So whenever cells divide, telomeres got shorter. Another problem, whenever you have DNA damage, especially double-strand break in telomeric sequences. For example, if you get DNA damage within chromosome, especially some coding sequence, if it's not fixed, cells most likely gonna go through apoptosis or get arrested or gonna die. If it gets in telomeric sequences, it's just gonna perceive as a normal telomeres, just short. So cells have way less division after that, because even just one telomere is enough, like when it's short, to stop cell division. Okay, so whenever cell, like length of the telomeres is critically short, cell becomes senescent, stop dividing, it's a fully senescent, it started to have, to excrete inflammation cytokines and some other problems, so creating niche, and I think a lot of problems actually connected to this process. So cells have specific enzyme called telomerase, which can increase telomere length. But in adult tissue, mostly in adult tissue, it's repressed. And the reason for this is a cancer. For example, you need several mutations to get cancer cells. And one of the protective mechanisms is this telomere, so telomere length. So for example, cells got pretty close to be cancerous, started divided. However, as soon as telomere short, it stops. So you're not getting cancer. And if telomerase or through some other mechanism it's reactivated, you can get long telomeres again, then those cells can become immortal and cancer. So this is one of the protective mechanisms how to prevent cancers. So in some tissue, telomerase is partially reactivated, like especially skin tissue or like some T cells or some other cells which require a lot of division. So it's reactivated to some level, but not fully. So telomeres in those cells still getting shorter. So we been thinking how to address this issue in those cells, and we knew that we can rejuvenate cells through induced pluripotent stem cell step. So it was many talks from me or Anya or any people on the campus how induced pluripotent stem cell make, but briefly, you kind of overexpress several factors in somatic cells like skin cells and create induced pluripotent stem cells. And they behave and look like embryonic stem cells. So they can divide indefinitely, they can be differentiated into tissue of interest. And one of the important, interesting things that they can be actually rejuvenated. People check the epigenetic marks, so they wiped out, those senescent marks wiped out. The telomeres get increased, but not right away. it requires up to 10 passages to get telomere lengths back to neonatal level. So in our experiments, in this specific experiment which I'm presenting, we took fibroblasts from, from 50-year-old patient, also 2 neonatal lines, FN2 and FN1. One that specific FF50 was senescent by serial passages. We kept almost a year to get it almost fully senescent. And we also took RDEB, our skin disease fibroblast. And as you can see, senescent fibroblast in this case actually clustered together on this RNA-seq data to RDEB fibroblast. And all those neonatal are more or less young fibroblasts cluster in different area. So whenever we created iPSCs from all of them, and you could see this is 2 iPS lines from every fibroblast lines. And as a control, we have 2 embryonic stem cell lines, H9 and H1, and they're all clustered together on this RNA-seq data. So we don't have difference or not much difference in iPSCs generated from younger fibroblast or senescent fibroblast. And then we also perform directed differentiation of 2 of these lines into fibroblasts. So one line was, again, fibroblast from this younger form. And again, we got fibroblast, we got iPSCs from them, and then we got back fibroblasts from them. And also we took the senescent fibroblast, generated iPSCs, 2 lines, and again, we generated fibroblasts. And as you can see now, these fibroblasts generated from iPSCs clusters together, way far from senescent fibroblast original. So suggesting that we can rejuvenate through iPS step. So question was, can we do it without making iPSCs? Because making iPSCs is quite a complex process, takes several weeks, overexpression of factors. FDA has a lot of questions if you try to use it in translational medicine, especially like tumorigenicity and other stuff. And especially big problem is differentiation. For example, again, I'm gonna show a small example. In our research, we're trying to make skin cells from iPSCs, keratinocytes and fibroblasts, and it takes months. And we spent years, again, getting this process working, and finally we arrived to making them through organoids, which sounds complex. So question is, can we make this fibroblast young again, expand them indefinitely, and keratinocytes, and even perform gene targeting on them without going through iPS stage and differentiation stage? So to do this, we decided to check what can be upregulated in iPSCs, why they rejuvenated that quickly. So one obvious target was, of course, telomerase complex. And telomerase complex is a protein complex which consists of 3 major parts. So this is a minimal complex. It's a H-TERT, called protein part of telomerase complex, H-TERC, which is the RNA component of RNAs, and DKC1. So mutation in any of them usually leads to some aging, like a disease which can recapitulate aging quickly, or some other degenerative diseases. And what a way how we can get this complex working in those senescent cells. Sorry, it's a little update too. So again, this is minimally complex, but we also have a lot of additional proteins which can be connected or work in tandem with telomerase complex, like TKB1, JR1, or some other one. And some proteins, additional proteins, which are connected to telomeres. They protect telomeres and from exonucleases, so like RAP1, TIN2, TRF2, TRF1. So those are proteins of interest if we try to do some like increase in telomere lengths and rejuvenation of those cells. And again, we reanalyzed our RNA-seq data, and we specifically checked, compared level of expression of those specific RNA transcription in fibroblast versus iPSCs. And as you see, many of them going up, like 3 times, 4 times. Okay, those are proteins, or in this case, RNA of interest, like trying to make a rejuvenation cocktail. But again, how we can do it? So we can use synthetic modified RNA. This is quite known now because it's used in vaccines, but overall, we have DNA template plasmid, which has T7 promoters, so we can make RNA in vitro, so in the laboratory. We can put any open reading frame of interest, like telomerase or any other gene. And we can put again UTRs, which untranslated region, and also poly-A, like in our case, it's 85 nucleotides, and it protects RNA from exonuclease to some extent. Also, when we synthesize it again, we have CAP analog on 5' end. It's also protected from exonuclease, and it's also responsible for entry into ribosome. So whenever we try to make a protein from this RNA, we need this one. However, it's my old experiment when I started back in immunology, when I was an immunology postdoc. I tried to make this kind of RNA, and I tried to transfect them to primary cells, not cancer cells, but primary cells. So results was that I lost all culture, so just wiped out. So later it was a paper that most of RNA in human are modified. And if we change some of nucleotides, for example, cytidine to 5-methylcytidine, just small chemical change, and same with uridine to pseudouridine, we can make this RNA less immunogenic. So cells are not dying as much. So we can transfect primary cells. Again, when it's a cancer cells, it's not usually a bigger problem. And similar modifications, I believe, used in Pfizer vaccine. So again, for COVID vaccine. So we can provide cells with a protein part, H-tert or any other part which is a protein. So we just put RNA into cells, it's translated, we have proteins. However, what we can do with TERT component, RNA component, technically we cannot make it in the lab easily because it has modifications, and modifications quite often in specific places. So unless you chemically synthesize it, it's difficult to do it. And if you synthesize it chemically, I don't think we can do it such a long RNA can be synthesized chemical. So usually it's a short RNA. Also, it requires H-tert to be present for correct folding and correct actual folding in telomerase complex. So we decided to check first, do we need it? Because we know that TERT usually present in somatic cells to some level. And we compare level of TERT in fibroblasts, 2 lines, one is 50-year-old and second is neonatal, versus 2 iPSC lines, which we got from the same line, and also embryonic stem cell line. As you see, we have increase in level of TERT several folds in iPSCs. So clearly we need it. So if we can increase, it would be really nice, but how we can do it? Because we cannot make it in the lab. So to avoid this issue, we actually used Cas9 VPR. So this is again another technology, which is many people are using right now. So Cas9 is usually bind to specific place in genome and cuts it. But in this case, we're using that Cas9, this is why it's D, and it's fused to VPR. VPR is a domain which can activate transcription from any area which you want. And this specific example from the paper we took it, it's actually activates MIAT or NeuroD1 or these 2 genes, and you can see that activation level from 280x, 87x, so 18,000x and 4600x. So, okay, we decided, can we use this molecule? And we made RNA encoding this full Cas9 VPR. And next, we took guiding RNAs, which are going upstream of TERT gene. And we try to find out which one can activate an increased level of TERT in cells if we apply both guiding RNA, which target to specific genome, and Cas9 VPR. And again, on these results, you could see that if we don't have anything, no transfection, it's one level. If we just put Cas9 VPR with without guiding RNA, we don't have effect. And then we have 15 different guiding RNAs. So first 4 are mixed together. So the best results we have with guiding RNA number 5, which is located right here. So some original experiments were done with mix of guiding RNAs, but later experiments we switched completely to guiding RNA number 5 because it's the best and we have best results with it. So first of all, we decided to check, okay, do we have telomerase activity increased in those fibroblasts if we apply this whole cocktail to cells? So here you can see telomerase SN, and if telomerase is present in cell lysate and it's working, you could see this increase in those fragments. So it means that like the sequence is synthesized. And if you hit your lysate, it means that any protein activity is killed there. So this is your actual control. So this is a control from KIT. And if we check F50 cells, so those fibroblasts from 50-year-old patient, this is how it looks without applying cocktail. However, just apply TERT only. We have telomerase activity going up, and we have apply full cocktail TERT plus DCAS9 plus guiding RNAs. We have it looks like it's even. And again, as a control, we have iPSCs. So you see the telomerase activity in iPSCs as well. So, okay, it looks like our RNA cocktail, at least TERT, DCAS9 VPR, and guiding RNA, it works. So we decided to check again, if it works, do we have other changes which we expected? And we decided to test telomere length increase. So again, we took iPSCs as a control generated from the same fibroblast. We took cells which we call senescent, but they're not fully senescent. There's still several doublings, usually 2 doublings before they're senescent fully, before they stop. It's just difficult to get them to that stage. It takes quite a lot of time. So this is telomere length as measured by qPCR as compared by iPSCs. So you see in iPSCs it's way longer compared to senescent cells. And if we treat these senescent cells with our cocktail, telomere length increase pretty high. So same with keratinocytes. These are not senescent keratinocytes, but it's late passage. If you try to senesce them, they actually start to differentiate. And again, if we compare to iPSCs, and again, this late passage is keratinocytes, telomere length is smaller than iPSCs. After treatment, it goes up. Finally, we tried to use MSC cells, so mesenchymal stem or stromal cells. And again, same. Compared to iPSCs, it's going down late passages, and after treatment, it goes up. So suggesting that our telomerase complex is not only active, but it's also increased telomere length in treated cells. In addition, we tested the amount of mitochondrial DNA. And again, some data that amount of mitochondrial DNA is going down in age in senescent cells. Same again here. We have lower amount of mitochondrial DNA, and after treatment it goes up. Same with keratinocytes, it's quite low. After treatment it goes up. And same with MSCs. Again, after treatment with our cocktail, consistent with these 3 factors, going up. Okay, these were original data, but recently we decided to switch to more clinical oriented protocol. And the reason, because we again working with iPSCs, trying to make skin cell therapy, and we're working a lot with GMP facility. And just not to make our life later difficult, we just decided, okay, this is still time we can go back and use GMP compliant components and try to make our protocol GMP compliant right here. So in all those experiments I presented, we used FBS, which is a fetal bovine serum. And it's very difficult, not very good idea to use in GMP compliant protocols. So we switched to human serum and some other modifications which we did to make it more GMP compliant. And this is our recent experiments, which I think very interested. So Oksana Breus, who's an instructor in my lab, just performs this, and she's leading this project right now. So she got cells fully senescent. So whatever I have shown is, let's say, 2 doublings left. These cells are fully senescent. They're not divided anymore. And it took quite a lot of time and effort to get them. So if we use beta-galactosidase staining, you see that they're 100% positive. And you see on this graph, and controls are positive. And nuclei, you could see also the interesting shape, like with DAPI. However, if we treat them with telomerase, only telomerase, not a full, we don't see much changes in this case. Again, we don't see new populations, almost all beta-gal positive, and as you see, there's no significant difference between control and telomerase treated, TERT treated cells. And if we use TERT for subsenescent cells, not full senescent, it does work to some extent. Just this, try to clear it up. However, if we use a full cocktail, I mean telomerase, DCAS9 VPR, and guiding RNA, so trying to activate both components, TERT and TERT, now we can see that part of our cells, about 50%, are not beta-gal positive anymore. And if we use EDU staining, EDU staining, which actually stain dividing cells, you can see appearance of this population from these fully senescent cells in our dish. And you can see even difference in nuclei shape or morphology. So it's interesting because it was reported that shape of nuclei is changed when cells are senescent. So it looks like it's going back. So they're rejuvenated even in this regard. And again, if we check number of senescent cells, beta-gal positive cells, it's 50% now because we still have some cell senescence, but usually they're gone with passage. We try to passage those cells and we're losing those senescent cells. And if we check number of dividing cells, it's 0 technically in control and only treated cells. And we're going up to 50%, a little bit more with cells treated with a full cocktail. And if we speak about morphology, we also can see how it looks. For example, on this left panel, you can see large senescent fibroblast. And whenever it starts, like whenever it's rejuvenated or cocktail is working, they become smaller and this nice appearing fibroblast. Similar on this middle panel, again, huge senescent fibroblast and smaller cells. And it's interesting that whenever you treat the senescent population of fibroblast, it's appearing in foci. So most likely some cells can go through this process and making these cultures of rejuvenated fibroblasts whenever we have some senescent cells left in culture. But again, as I told, they're usually gone with pathogen. So this is results from this experiment. So we try to see how many population doublings we can get from these cells after treatment. And here we have several important controls. So one control is just DCAS9 and guiding RNA only. So we don't have third component here. As you could see, we don't have proliferation. Same with the control, the cell transfected just with wasabi. If we use telomerase only, we do have some expansion of cells, but it stopped going on this plateau again. If we use dominant negative form of telomerase, this is an interesting mutant which has all activities of human TERT or telomerase, but cannot increase telomere length. So this is mutated, 2 amino acids mutated, and it cannot increase telomere length, but it can do all other stuff. But again, in our case, as you can see, we need telomere length increase, otherwise it's not working. And again, our cocktail gives expansion to cells and they're going through expansion. So this is our data with this cocktail. However, we decided to go forward, and I'm sorry for such a complex figure, but we decided to test many other factors which have been reported, which we think can work. And this is results of experiment as of today. So it's still in progress. And this is, again, today's technical as yesterday results, but it's still in progress, and we would like to see what's going to be then, how long they can be in culture, how long they're going to divide. So again, so this is lower light. It's one of the cocktail which was previously reported. So, and it was used not on senescent cells, just on like middle cells. And as you see, it doesn't work in our conditions. This is our control. Also, like you don't see any increase in length. This is subsenescent cells, by the way. This is about 2 more passages, 2 more doublings left before it stops. This is telomerase only. So you could see it's how many doublings it went through, and it looks like it's going on plateau. Also, this is our cocktail. This is green line with the blue circles. This is cocktail which I just described. So telomerase, TERT, and guiding RNA DCAS9 APR. However, we tried different additional factors combinations or different combinations, and you see that it can increase. Especially one of the combinations, which I just call best combination here, it's, I would say, it's about 47 versus 52 doublings for the same amount of time. So we have 5 more doublings within this period of time with this combination. And it looks like it's still in exponential phase. So, okay, we got these cells, and it's interesting, they can divide. We have some telomere, mitochondria, but what about their functionality? So can we see that the function improved? And one of the assays we performed, and thanks to Michael Ferreira's, it's a transendothelial migration assay. For this assay, we have 2 plates, one in another, and inner plate seeded with vascular cells to mimic blood vessel. And on the top, we apply MSCs, which are GFP labeled. So we have GFP labeled MSCs, and microscope is underneath, so we can look underneath. And this is fluoroblock here. So we cannot see this MSCs GFP unless they're going through this membrane. This is tiny pores, so they have to go through those tiny pores if we want to see them. So we put mimic inflammation on the bottom chambers to attract those MSCs. And we can evaluate migration efficiency of those GFP MSCs. And what we found that if we compare early passage and old passage MSCs, the same MSCs, just a different passage, this is quite huge difference in the ability to move through those pores. So you can see on the left that young MSCs or early passage MSCs move faster through those pores and move around way better than late passage MSCs. So we did quantify it, and again, you can see that effect is quite significant between MSCs passage 5, we called young, and MSCs passage 14. So they're losing this ability with passaging. So we decided to take this aged late passage MSCs, treat them with our rejuvenating cocktail, and compare them to young and again original MSCs. And as you can see, we have appearance of this rejuvenated MSCs even before young MSCs appear, and of course, way better than late passage MSCs moving through. And again, after quantification, we could see late passage MSCs, their movement through the pores, young passage 5, and the same old or passage 14 MSCs treated with a cocktail move way faster through pores and around whenever we treat them with a cocktail. So another interesting characteristic of aged cells is a methylation profile. And again, this is very high profile, several very high profile papers saying that methylation profile is changing with aging. And what we decided to do, we just collected, again, cells with high passage culture. And it's again, this is all three types of cells, MSC, keratinocytes, and fibroblasts, low passage culture, and this high passage culture treated with our RNA cocktail. And we've used Illumina epic methylation assay, and Andrei Rajok, he analyzes data. And what he did, he just tried to combine all of them and find sites which are similar to between all of those cell types. And what he found that these specific sites which he found at low passage, their methylation is quite low. This is red line. However, at high passage, methylation increased up to 50% or 60%. However, if we take these high passage cells and treat them with our cocktail, quite often it's going down to the original level of low passage or somewhere in between. So suggesting that we have not only this functional assay, like with MSCs, but methylation profile, which is one of the also hallmark of agent, is also changed back. So another interesting experiment which we performed is a proteomic assay, and we specifically been looking for post-translational modification on cysteine. So cysteine has SH group, so it can be oxidized or it can be still in reduced state. And depending on different processes, again, it's oxidized. This was a very interesting paper saying that it changes in specific way in aged tissue. I think it was mouse. And we decided to analyze it on our cells. And Oksana did it. And we got very interesting results. So she compared oxidation level of those cysteines in senescent cells versus young. And you could see that specific examples that they're way more oxidized in senescent cells, like is in log 4, however, just 2 like in young cells. And if we take these senescent cells and again rejuvenate it with our cocktail and then perform analysis again, we can see that those specific proteins actually the oxidation level it changes. Again, in senescent cells, for example, it's here like this one, and in rejuvenated cells, it's way less oxidated, even less than in those cells we called young. And we found interesting ways. Quite a lot of proteins are more oxidated in senescent cells and then less oxidated in rejuvenated cells. But we also found proteins which behave different way. They're actually more oxidated in young cells and less oxidated in senescent cells. However, upon treatment with this cocktail, they revert and revert even more than in young cells. So we suspect that actually, because we call those cells young, but they're early passage. Probably when we treat them with our cocktails, they're getting even more push, like more neonatal level. So another functional assay, like very strong functional assay we decided to perform is to test if we can form a human skin on a mouse. And to do this, we're using one of assay which we use quite often in our lab. So in this assay, you can mix human keratinocytes and human fibroblasts, which you grow in culture. And then on the back of a mouse, you make a cut and put a like chamber in like with open facet. And then you mix those keratinocyte fibroblasts and put inside. So at some point you remove this chamber and you can have formation of human graft. So if those cells are capable, they can form on human skin on the back of a mouse. And this is how it looks. So the chamber, again, we put a mix of human keratinocyte fibroblasts there. Then at some point it's removed and you can see formation of human graft. So then after healing, the graft area can be harvested and analyzed. So in this case, we could see formation of human skin on the back of a mouse. And as you can see, it's more thick, more layered there compared to mouse skin. And if we stain with specific antibody, so one is keratin 1, which is stained mouse tissue, what you see here, so it's stopped here. And keratin 14, which stains both human and mouse. And this is specific for keratins. And you can see whenever human skin is formed. So again, we took senescent cells and tried to perform the similar procedure on the mouse. And in this case, it failed. And doesn't matter in which combination you use it. For example, if you use young keratinocytes and senescent fibroblast, you don't have a graft, or vice versa. If you use senescent keratinocyte or late passage keratinocyte and young fibroblast, again, you don't get human graft or human skin formed on the back of the mouse. And again, this is a mouse skin, it's stained pretty well with mouse, but we don't have any human component here. However, if we take those aged or senescent cells and treat them with our cocktail, and then we try to graft it, now we have formation of a human skin. So you could see, again, layers of human skin there. And if we stain it with keratin 1, this is where its mouse tissue stops, and human vimentin, which is a marker of dermis, so underneath part of this graft, it can stain pretty well. This is fibroblast here. And we stain again with keratin 14, which is, let's say, human in this mouse. Again, we could see formation of nice human skin. And with human loricrene, just to find out where is the human tissue, you could see staining here. So again, those cells which were functionally not capable to make a graft anymore, after treatment with our rejuvenation cocktail, could reproduce skin again on the back of a mouse. And I think this is, we think this is one of the most stringent assay for functionality of those cells. Okay, summary of part 1. So the delivery of our RNA cocktail, and again, experiments which I have shown for functional assay. This is done with our more basic cocktail, which include VPR, TERT, and guiding RNA for TERT. Right now, we're trying to get even better cocktail with additional factors, so we will need to repeat some of those experiments. So again, our basic cocktail increases telomerase activity in cells, increases telomere length. We also can increase proliferation potential of senescent cells, and in one specific case with the fully senescent human cells, we can restore them and divide again. And to my knowledge, I don't think it was done I didn't see any other report doing this. So it can increase mitochondrial DNA number, improves MSC migration, restores DNA methylation marks, as well as thiol group oxidation, and does not appear to affect the ability of keratinocyte or fibroblasts to form grafts. So one of the critique which we got from our grant, it was that they've been thinking about if we immortalize our cells, so is it any problem? So we know that telomerase is gone from those cells which we use for making graft, like human skin graft on the mouse cells. So again, we treated them, we waited until telomerase is gone, so we know it's safe. And then we generated graft on mouse skin. So now I want to switch gears a little bit and speak about our future steps and potential applications. So I will just concentrate on one potential application specifically. However, I will mention some other applications as well. So basic research, we would like to study connection between telomere length, epigenetics, and mitochondrial function. So again, it was shown the connection between the short telomeres, epigenetic, methylation profile, and anything else. So our question is, if we can increase telomere length back, and we know that telomerase is gone because it's RNA, so at some point telomerase is gone actually quite quickly, will we have changes in addition? So we've seen some changes as I just presented in methylation profile, but those methylation clock which people are using, and there's a specific methylation clock for skin is known. So do we have it back fully? Do we have changes in mitochondrial function fully? So we'd like to investigate this. So each of these pathways can be investigated pretty well, and this is a lot of work just if we want to show that those cells are similar to young cells. Of course, we have to check probably mutagenic load as well, just sequence them and see do we have any deleterious mutation accumulated, and is there any problem with this one or not. Same, we also can study other activity of telomerase complex, because again, we have this control which cannot increase telomere length, but have other functions. So we can see, is it doing anything? However, from our preliminary experience, we just have shown, looks like not. We don't see appearance of this population or proliferation of cells after treatment with dominant negative TER, dominant negative TER. So how we can use it for translational research? So one of this which we would like to work is adapt this rejuvenation gene targeting approach to the treatment of epidermolysis bullosa. And as you know, and this is our specialization, we're working with a skin disease. So right now we're working with the iPSCs. We successfully targeted them, we successfully changed like corrected mutation. We making skin cells. Skin cells are grafting, and Marina Pavlova are working on this. And right now we're moving it to GMP facility to perform a clinical, clinical experiment there. However, we hope we can use this approach too, and it may be faster and easier later on. But again, it probably requires several years. So we probably can use this RNA rejuvenation cocktail for generation of CAR T cell lines. And again, as I told in the beginning of my talk, that for aged people, T cells, it's a problem with the expansion because problem of telomere length, same with those tumor infiltrating lymphocytes. So maybe if we use our cocktail, we can restore function of CAR T cells and they may be more active. So this is one of research direction we want to try. And finally, we might try this RNA cocktail for MSC lines. So right now, there's several companies which try to generate MSC lines from iPSCs. So trying to get similar line and for many functions. Maybe with that cocktail, we don't need to go through iPSCs again. We can probably generate them from just lines and expand them as much as we need, of course, with proper controls. So again, I would like to concentrate on gene targeting and skin diseases and this approach. So one of the reports several years ago, 2017, as a boy was in Germany who lost almost all his skin. And this is how he looked. So he was dying. And Michele De Luca got permission to perform skin, like trying to correct skin cells and put them back on this poor boy. And what they've done, they took skin cells and used a virus to introduce normal copy of LAMP3 genes, which was responsible for this horrible disease. And then they expanded those skin cells and put them back on this boy. And later on, we spoke with Michele De Luca, and he told the boy was playing soccer and he was leading pretty happy life, at least several years after these procedures. So idea is that we can correct somatic cells and use them for this kind of treatment. And now we're going to RDEB. This is what we're working on. So RDEB usually is a mutation collagen 7A1 genes. And very simple, like simplifying, we have dermis and epidermis of like in skin, And those epidermis connected to dermis through basement membrane. And major component of basement membrane is collagen 7A1. So if mutation is there, this is what happened. So skin is falling off, blisters forming very quickly. And it would be nice if we can correct it. So previously, Zurab Siprashvili and the team in Stanford, they've been able to correct collagen 7A1 through lentiviral vector. They put it back into keratinocytes, grow those grafts, and put back to patient, at least have information of some basement membrane. So restoring this collagen 7A1. So whole idea why I'm showing this data just to show that it is possible to do on somatic cells. However, whenever you use them, again, you have troubles with expansion, especially RDEB cells. Again, if you first remember first of my slide, like one of my first slides, they're pretty close in the appearance to senescent cells, and you probably cannot expand them too much. So it's a problem. Again, we in the lab trying to solve it through iPSCs, but this is maybe another approach doing it. Okay, this is a strategy for how we try to correct it right now in the lab. So we again collect skin cells, making iPSCs, correcting those mutations, expand, differentiate back into keratinocytes and fibroblasts, and return them as a graft into patient. Recently, what we've done, we tried to simplify these procedures and try to reprogram and genetically correct this RDEB mutation at the same time. So we're saving a lot of steps and time for GMP facility. And again, how it is done. Again, we have this mutation, C to T, and we have repair single-strand DNA, we have Cas9 guiding RNA, which cuts close to this area, and then through homologous recombination, it's fixed. And we generate iPSCs. So applying Cas9 guiding RNA single-strand before reprogramming starts. So again, whole idea is that this correction is happening while cells still fibroblasts. So they're not iPSCs yet. So again, we generate then iPSCs, which you can see by TRA160 positive staining, and they usually derived from a single cell. So we took them out, establishing many clones, and analyzing them using ddPCR machine. And again, the ddPCR machine is able to perform many, like in one PCR reactions, you actually performing like 20,000 PCR reactions in oil. So this is all different. And depending on what kind of probe you have, green, for example, or blue for wild type, that little like droplets will be green or blue depending on which DNA molecule was there. And then you can actually quantify it and find out what is the ratio of blue versus green and what we have. So as a control, you could see how it looks. So this is a green is a full mutant with RDEB mutation, and blue is a wild type. So we tested and we analyzed those colonies, and you could see that we have some colonies which have both signals. And especially when it's close to 1-to-1 ratio, it's pretty nice. So it means that we have cells or APS clones in here with corrected mutation because our patients are homozygous for this mutation. So now we have wild type and we have mutation. Again, we did sequence this line. We found 3 different type of clones. So correction of one allele, unmodified, so no correction, or deletion, large deletion. Of course, we're going to use these iPS clones with one allele. And we actually been able to correct both alleles in some instance. So why I'm telling all of this? Because we can use similar procedures without making iPSCs. Again, this is our work, how it looks now. Can we do this? Can we just collect somatic cells, corrected it, rejuvenate it, and expand it, and return it as a graft without all those additional steps? And to do this, we performed 2 experiments just as a preliminary experiments. In first case, again, as I told, we've been suspecting that we don't have any problem in correcting fibroblasts directly. And on the left part, those AB, this is RDEB fibroblast. Very similar strategy, again, blue-green probe, and we just try to correct fibroblast. And after analysis, we found that we correcting about 5% alleles. Since it's homozygous, close to 10% of cells are corrected. So next, we moved and tried to correct keratinocytes because this is 2 types of cells. In this case, we need to correct them both. And again, if it's only Cas9 single-strand DNA but no guiding RNA, we don't have any correction. But if we have all components, we actually can correct about 8.9 or 9% of keratinocytes in population. So idea now is that if we can seed them for single cells, grow enough cells from single cell using our rejuvenation cocktail, because it's very difficult to grow anything if they quickly just stop growing. We can then identify those cell lines which got corrected and use them for grafting procedures, first one of mouse, of course. And to do this, we just took adult fibroblast and adult fibroblast treated with our rejuvenation cocktail and seeded them as a single cell. So as you can see, we generated, I believe, only in two cases, we could see division of cells and getting some fibroblast in those lines. However, if we applied cocktail to the same cells, now we have 8 or 9 wells with growing fibroblast population. So this was one of the data we generated. So this is the end of my talk, and I think that again, we probably one of the way we can apply this rejuvenation cocktail is combined with gene targeting, especially for some monogenic diseases. And I would like to thank our laboratory and especially Anya, who worked with me on this project from 2012, like a long time ago, when we tried something and then it stopped. And when we had some findings, we continued. So previous person was Nicole Dieta, so she is here. So she worked a lot, and Sean too, who helped to generate some construct. And then this project, Nicole left, and she's assistant professor right now. And Sean left too to another place in the university. And then this project moved to Oksana, who generated most interesting data recently. And she's working with Macra and with Jasmine right now. So generating all this interesting data. Also thanks to Michael, who generated all the MSC's data. Andy Rajok, who generated all performed all bioinformatic analysis for our, and also everyone else in the lab who helped, like Laura with histology, and Kalpana culturing those cells, and many other people who in the lab help with this. Also, I would like to thank our IP and administration support, and especially Heather, who helped us to submit a patent, Aoife, who is working with our patent right now, Carmen Garcia for her administrative support with all this research, of course, Dennis for leading Gates Center and helping with all those projects, Department of Dermatology, Dr. David Norris as a chair, Children's Hospital Colorado, Anna Bruckner, who works with us, and we're working, like, we're getting patient samples working with her, as well as my finding from NIME, Sarah one, which I got, and TCD2 grant to Dr. Norris and Dr. Root. And recently, huge thanks to Gates Frontier Fund, especially Gates Grafts Fund and Diane Gates Wallach, specifically for this generous award, which now we can use to go with this project further. Okay, thank you very much, and please, questions.
That's good stuff, Igor. Thanks for sharing. I had to step out for a minute, so I might have missed this, but how long does the reprogramming or the rejuvenation mix persist in the cells, either at the RNA level or the protein level? Do you know?
Not very long. So again, this is our recent experiment. This is just several days, and we've checked telomeres activity and presence, mostly on telomeres activity. Yes, it's just not long. This is why we have to, I mean, this was part of experiments. We've been looking how long we have to do those transfections. This is not one transfections, it's multiple transfections. So we did, right now, I think we stopped at 4 transfections, and I believe it's every several days, yeah. But at some experiment, back at Nicole did it with, again, FBS, and this is different now. She treated them for almost 2 months. So